All right, we left off in the last video towards the end of bacteria, and we are going to go ahead and get my, there we go. We're going to go ahead and finish bacteria and start protists in this video. So we left off, we talked about the fact there were three domains. We have bacteria, archaea, and eukaryota. Eukaryota has membrane-bound organelles. It has a nucleus. They're larger. Bacteria and archaea are both bacteria. Sometimes bacteria is called eubacteria, EU bacteria. Let's spelled with an EU. But what we see is that archaea, pen's not working, and eukaryote are actually more closely related. So we have a table here. Before we get to that, a lot of archaea are what we call extremophiles, where they live in conditions that are not hospitable for other organisms. So if you're a halophile, you are living in a high salt concentration. A metallophile is metal, thermophile is heat, psychrophile is cold, acidophile is acid, and alkylophile is an alkaline or a basic situation. So these guys can go places other organisms can't. But here's what we see. So if we look at what is the same and what is different between bacteria, archaea, and eukarya, let's see if I can switch to a highlighter. Oh, that is not what I wanted. Give me just a second here. Highlighter. All right, so if we look, you'll notice that peptidoglycan in the cell wall. Bacteria have it, but archaea does not, and obviously eukarya does not have cell walls. So RNA polymerase, again, here's a similarity between archaea and eukarya. Introns, another similarity. And finally, antibiotics. So we have all of these things that are similar between archaea and eukarya that aren't the same between bacteria and archaea. So we actually believe that archaea and eukarya are more closely related. We talked about whether or not a bacteria could live in oxygen. So we said obligate anaerobes here. They will die if they're exposed to O2. An means without, and aero is oxygen. If you're an obligate anaerobe, you have to have oxygen to live. Now, facultative anaerobes and atolerant anaerobes, these guys can exist in conditions where we have a little bit of both. And you notice here I've got my high O2, and down here I've got my no O2. So that kind of shows you who can live where. So these were 4 definitions. It's good to go back over the meanings for all of these. Again, just going to hit the high points, not discussing in detail. We talked about, oh, that should have a Y up there. Switch back to a pen. So, oh, I'm still in highlighter. Sorry about that. Give me one more second. Pen. Okay. So we have, there's another group here that we don't have listed, and that's proteobacteria. So we've got cyanobacteria, we talked about these guys probably the most because of their role in photosynthesis and creating oxygen. Chlamydias, these are gram-negative. They are parasitic in humans. We had spirochetes. These are also gram-negative. So this is gram-negative. This is gram-negative. Down here, gram-positive are their own group. And proteobacteria, we're going to see, they believe these are also gram-negative. They believe these had a part in the endosymbiosis for the first mitochondria. We talked about the roles that bacteria have. They can be mutualists, they can be parasites, they can be commensal. But decomposition is kind of considered its own thing. So we said that being a decomposer was its own niche. And we had examples here of parasitic bacteria. We have growth bacteria in the soil that assist growth. We have bacteria in our guts that's mutualistic. Again, sometimes it is the fact that it benefits the bacteria and it doesn't hurt the host. So we have a wide range of bacterial ecological niches. We did exotoxins and endotoxins. Exotoxins are produced inside the bacteria and then released. An endotoxin, remember those lipopolysaccharides? Often it's not always the lipopolysaccharides, but it's very commonly the lipopolysaccharides. And if this bacteria is killed and it breaks up, what we have here are those lipopolysaccharides that get released into the host if it's parasitic, and it's usually toxic. So that's an endotoxin, exotoxin. Then we talked about antibiotics and some of the roles of bacteria. We're not going to focus on that too much. Then it was time to go to protists. So we said that protists, the one thing that every protist has is a mitochondria. So the requirement there, you got to have a mitochondria. And the use of kingdom Protista is a little bit outmoded. We talk about it more in terms of supergroups, but from a teaching perspective, it's not bad to talk about all of these mostly. And so this is all, but most are single cell, not 100%, but for the by and far, 95% of all the organisms in Protista are single cell. We did a quick review of organelles. We're now into eukaryotic cells, and eukaryotic organelles are much, much more complex. So then we started to talk about endosymbiosis, and here is that proteobacteria. So we have a archaea. They believe that the recipient or the host was an archaea bacteria, and they think the one that was engulfed was a proteobacteria. They believe this was a singular event. For plastids, it happened more than once, but for mitochondrial endosymbiosis, they believe it really only happened at one point. And our evidence is that this guy has its own genetic material, and it has the membranes that are, so it shows here, like this is the host membrane, and then I have the bacterial membrane. So the cell wall was lost over time, but you can see we still have two membranes. So because of the arrangement of the membranes and because mitochondria have their own genetic information, that's pretty strong proof that this is where mitochondria came from. Plastids are things like chloroplasts, leucoplasts, chromoplasts. We have rhodoplasts. We've got more than one type of plastid, even though the only one you probably have heard of before now are chloroplasts. So we had initial endosymbiosis where we had, now in this case, we had a cyanobacterium, and we're now going into a eukaryotic cell. So this guy's already got a mitochondria. So they believe that the mitochondrial endosymbiosis took place before plastid. Again, the chloroplast, so here we're being engulfed, and we end up with multiple membranes, and this has its own genetic material. And that's pretty strong proof that this is where chloroplasts came from. They believe then that eukaryotic cells with plastids, with chloroplasts, or in this case, so this would have chloroplasts down here. This guy being red would have a rhodoplast. So this is the initial, the primary endosymbiosis, where we have the eukaryotic cell taking in the cyanobacterium. And in this case, it would have been a slightly different one because it has a red plastid to it. But what they believe happened was then, so this is what the result was: a red alga or a green alga. They think the red alga and the green alga were then engulfed a second time, and we call that secondary endosymbiosis. And what that did was it created a structure that not only has its own genetic information, but you see this little dot down there? It's called a nucleomorph. And that nucleomorph is a rudimentary vestigial nucleus. So we see it in the stromatophiles, the alveolates, euglenids, and we really focused on the fact that it happened with euglenids because we talked about euglenids quite a bit. Here was the larger tree. I'm going to go ahead and go forward so that we're oriented in the right direction. So we had things that were boxed in green and we had things that were boxed in yellow. And the things that were boxed in green are the ones that we spent more time on. We mentioned that the parabasalids and the diplomonads have primitive mitochondria. They can't use oxygen to create ATP. So we just mentioned that with them, and that's not going to show up on the final. The euglenozoans, we talked about euglenids and kinetoplastids. So the kinetoplastids, that was African sleeping sickness, the tsetse fly. And euglena, then we spent, I think it's got its own page, so I'm going to wait on that. Diatoms have its own page. Brown algae has its own page. Dinoflagellates do. Apicomplexes do. Paramecium do. So the radiolaurans and the forams and the cercozoans, again, we talked about those briefly, but because we didn't focus on those, I'm going to skip those in the review video. Our first one that we spent a little bit of time on were euglena. They're in excavata. And by the way, I forgot to mention, so here are those super groups. I'm going to go back here. We have excavata, we have SAR, we have archaeplastids, and we have uniconta. So really, this is the way they talk about protists, are these super groups. And with the euglenids, we talked a little bit about their anatomy. We talked about this contractile vacuole. It allows them to exist in situations that are more hypotonic or hypotonic because they can pump out. Actually, it would be hypotonic because they can pump out fluid. They have a rudimentary light detector. They have two flagella. So they've got a short flagella and a long flagella. And so that's one of the things we look for euglenid. They're pellicle. These are like armadillo scales that are layered. We said too, these guys can be mixotrophs. So it's important to remember the nutritional profiles for all of these protists. So they can be both photosynthetic or heterotrophic. Some of them are actually predatory. Diatoms, these guys are they're also known as phytoplankton. Because they are the most important primary producer in marine situations. And they also, when they die, they tend to take all that carbon with them and they sink it to the bottom. So they're a carbon sink. If you look at soil that has dead diatoms in it, we call it diatomaceous earth. Their big unique aspect are, if you have a hard shell, it's called a test, and their tests are made of silica. They float. They do not have directed motion, so they are considered planktonic, means you float, you don't have directed motion. We looked at meiosis in humans because we do a lot going forward with life cycles. So we reviewed meiosis. Go ahead and do that if you want. We talked about the first organism we talked about, and we got into a pretty detailed life cycle, was brown algae. We did the anatomy of brown algae, where we have the blades, we have the float, we have the stipe, and we have the holdfast. So that allows and they each have their own function. But this is where we really started to break things down. So we have alternation of generations. We have, and in pink, we have the diploid form, and in blue, we have the haploid form. So what we see here was we had fertilization, and that brings together an egg and a sperm, and the from fertilization, you always get a zygote. Now, as a cell divides, it stops being a zygote very quickly. We consider zygote to be that fertilized egg. And we have growth and development until we end up with the what you would think of if you looked at an algae, you would say that this is brown algae. But what actually happens is we have these sporangia. These produce haploid spores. Oops. Called zoospores. Those zoospores differentiate into female and male, and they're very, very small. So you don't see them, but these are actually considered to be their own living organisms. So that's why we call it alternation of generations. You have um, the diploid structure, the sporophyte, and you have the haploid gametophytes. So this is the one organism, and this is another. The gametophytes, uh, the zoospores differentiate into male and female gametophytes. And it's here when we go through the fertilization process and we have the male and female, this is our source for variation. And one of the things, every time we talk about a life cycle, right, we have to talk about where does that variation come from? Because we know that natural selection can't work unless you have variation. So variation is super important. And I think that's everything we had for alternation of generations for brown algae. This is probably a good stopping point. So my last video, I will finish protists and we will do fungus. All right.